Welcome to this lecture on the host-parasite relationship. Imagine having a roommate who never pays rent, eats all your food, and makes you physically sick. In the natural world, this ultimate bad roommate is called a parasite. Today, we are going to explore this fascinating and often destructive dynamic. We will learn how parasites rely on their hosts, the different roles hosts play, and the mathematical tools scientists use to track and predict parasitic outbreaks. Let us dive into the dynamics of survival. To begin, let us define exactly what we mean by a host-parasite relationship. In scientific terms, this is a specific type of symbiosis where one organism, the parasite, lives on or inside another organism, the host. But unlike mutually beneficial relationships, this one causes harm to the host. The parasite is metabolically dependent, meaning it cannot survive on its own. It steals essential nutrients, uses the host as a habitat, and relies on it for a safe place to reproduce. Meanwhile, the host's immune system constantly fights back, creating a never-ending evolutionary arms race where both species are constantly adapting to outsmart each other. Now that we understand what a parasite is, let us look at the victims of this relationship. Hosts are not all the same. They are categorized based on the specific role they play in the parasite's life cycle. First, we have the definitive host. This is the organism where the parasite finally reaches sexual maturity and reproduces, ensuring the continuation of its species. On the other hand, we have the intermediate host. This host harbors the parasite during its crucial developmental or larval stages before it is ready to move on. Understanding these distinct roles is essential for figuring out how a parasite spreads through an ecosystem. Building on those definitions, let us visualize this journey with a diagram of the transmission cycle. On the left, you see the human, acting as the definitive host. Inside the human, adult parasites reproduce. Following the arrows downward, you can see parasite eggs being released into the environment, often near water sources. These eggs then infect the intermediate hosts, shown here as a snail and a mosquito on the right. Inside these intermediate hosts, the parasite undergoes crucial larval development. Finally, the top arrow shows the transmission back to a new human host, completing the cycle. This visual map helps scientists identify weak points where we can intervene and stop the spread. Having seen how parasites cycle through hosts, you might wonder why we care so deeply about tracking them. The answer lies in global health, agriculture, and ecological stability. Millions of humans and animals suffer from devastating parasitic diseases like malaria, schistosomiasis, and tapeworm infections. These diseases cause massive economic and health burdens worldwide. In agriculture, a parasitic outbreak can wipe out entire livestock populations, leading to severe food shortages and economic collapse. By mastering our understanding of this relationship, scientists can develop targeted drugs and vaccines that disrupt the parasite's life cycle without harming the host, ultimately saving countless lives. While treating individual patients is important, to truly understand the impact of a parasite, we must zoom out. We cannot just look at one individual. We must measure the infection across the whole community. Epidemiologists do this by translating biological observations into hard, actionable mathematical data. Without these mathematical tools, it would be impossible to know if a parasitic outbreak is getting better or worse over time. We rely on two critical mathematical metrics to evaluate the burden of disease in a population: prevalence and mean intensity. Let us explore the first of these metrics. This brings us to our first mathematical tool, the formula for prevalence. Conceptually, prevalence tells us how widespread a parasite is within a specific group. The formula is P equals N sub P divided by N sub H, all multiplied by 100. Let us break down these symbols. P represents the prevalence, which is a percentage. The variable N sub P stands for the number of hosts that actually have the parasite. The variable N sub H represents the total number of hosts examined in the population. The multiplier of 100 simply converts the decimal into a readable percentage. Physically, if the number of infected hosts increases while the total population stays the same, the prevalence goes up, indicating a rapidly spreading infection. Before we move on to the next metric, let us see this formula in action with a concrete example. A public health team visits a rural village to study a hookworm outbreak. They examine a total population of 500 villagers. Out of these, they find that 150 villagers are infected. We need to calculate the prevalence. We chose this formula because we want to know the percentage of the village acting as a host. We substitute 150 for the infected hosts and 500 for the total population. Dividing 150 by 500 gives us 0.30. Multiplying by 100 gives us our final answer. Physically, this means that 30%, or nearly 1/3 of the village, is infected with the parasite. Now that we know how widespread the disease is, we need to know how severe it is. This is where the mean intensity formula comes in. Conceptually, this calculates the average number of parasites living inside a single infected host. The formula is I equals P sub T divided by N sub P. Here, I represents the mean intensity. The variable P sub T is the total number of individual parasites collected across all infected individuals. And N sub P is the number of infected hosts, exactly as we used in the previous formula. If the total parasite count increases, but the number of infected people remains constant, the intensity increases. Physically, this means the infected hosts are getting sicker because they are carrying a much heavier parasite load. Let us continue with our hookworm example to find the mean intensity. After administering deworming medication to the 150 infected villagers, the medical team collects and counts a total of 4,500 adult hookworms. We want to know the average burden on the sick individuals, which is why we use the intensity formula. We substitute 4,500 for the total parasite count and 150 for the number of infected hosts. Dividing 4,500 by 150 gives us 30. This final answer tells us that each infected person is carrying a significant burden of about 30 worms. This high intensity could cause severe anemia and malnutrition if left untreated. Beyond measuring current infections, epidemiologists also need to predict future outbreaks. They do this using the basic reproduction number, denoted as R0. Conceptually, this represents the average number of new host infections generated by a single infected host in a completely susceptible population. The formula is R0 equals beta times C times D. Let us define these variables. Beta is the probability of transmission per contact, such as a mosquito bite. The variable C is the contact rate, meaning the number of bites per day. Finally, D is the duration the host remains infectious, measured in days. If the contact rate or the infectious duration increases, R0 goes up, meaning the parasite will spread much faster through the population. Let us apply this predictive formula to a real-world scenario involving malaria. We are given a transmission probability of zero point 0.2, a contact rate of 5 bites per day, and an infectious duration of 10 days. We are asked to calculate R0 to determine if an epidemic will occur. We use this formula to combine biological infectiousness with behavioral contact rates. First, we multiply the transmission probability of zero point 0.2 by the contact rate of 5. This gives us 1.0, meaning there is an average of 1 successful transmission event per day. Next, we multiply this daily rate by the 10-day duration. Our final answer is 10. Physically, this means one infected host will pass the parasite to 10 new hosts, indicating a severe epidemic is imminent. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that the host-parasite relationship is a complex battle where parasites extract resources at the expense of their definitive and intermediate hosts. Second, we saw how mathematical metrics like prevalence and mean intensity allow us to quantify the exact burden of a disease on a community. Finally, we used the basic reproduction number to predict how rapidly an infection will spread. By mastering both the biology and the mathematics of these interactions, medical professionals can break the chain of transmission and protect vulnerable populations. You now have a solid foundation in the dynamics of parasitology.